everyone. In this class, we'll try to learn about neuron. Neuron is a structural and functional unit of nervous system. The term neuron is used to describe the nerve cell and its processes, the dendrites and the axon. If you see the structure of the nerve cell, it carries, it is made up of cell body, that is soma or perikaryon. There are various sizes and forms. They can be stellate, round, pyramidal, or fusiform. Its principal constituents are similar to a generalized cell. However, after fixation with the special stains, its cytoplasm also reveals presence of nacelles, granules, or bodies. These are the basophilic granules composed of many thin, parallel arranged membrane-bounded cavities or cisterns, which are covered by many minute particles consisting of ribose nucleoproteins, that is RNA, with proteins. Granule size and number varies with physiological condition of the cell. Neurofibrillae, these are the fine threads 6 to 10 nanometers in diameter and of variable length. They traverse the cytoplasmic matrix, forming a loose framework of fibrils in the cytoplasm. There is no centriole, which indicate that the highly specialized nerve cell lost its power of division. Nerve cells, once destroyed, are replaced merely by neuroglia cells which supports the nerve cell. Dendrites. There are 5 to 7 processes extending out from the cell body and arborize extensively after they leave the cell. They also contain nacelles, granules, mitochondria, and neurofibrillae. They are the receptive processes of the neuron. Impulse can be transmitted from one dendrite to another in the central nervous system. Axon, it's also called as axon cylinder or nerve fiber. It originates from the thickened area of the cell body, which is called as axon hillock, in which there is no nacelles granules. Cytoplasmic fluid occupying the center of the axon is known as axoplasm. The cell membrane enveloping the cytoplasm is also continued on the axon as axolemma. Axon vary from few microns in length to as long as 90 centimeters. Axon is a single elongated cytoplasmic extension with the specialized function of conducting impulses away from the cell body. Synaptic knobs, they're also called as terminal buttons or axon telodendria. The axons divide into terminal branches, each ending in a number of synaptic knobs. They contain granules or vesicles in which synaptic transmitter secreted by the nerve is stored. Myelogenesis, neurolemma or sheath of Schwann has got a cell, Schwann cells, which takes part in the deposition of myelin sheath around the axon, a process called myelinogenesis. Myelination of axons in the central nervous system is by the oligodendrocytes. One oligodendrocyte processes up to 40 axons and forms the myelin around them. So this is structure of typical neuron, consists of cell body, axon, and terminal buttons. You can see the fine the granules, nacelles granules, and the axon hillock, the axon which are covered by the myelin sheath. In between you can see the node of Ranvier and the terminal buttons. Glial cells, they are also called as neuroglial cells. Glial means glue. These are the cells which support the nerve cells. Glial cells are very numerous. There are approximate 10 times as many glial cells as neurons. Unlike the neurons, glial cells are capable of multiplying by mitosis. The glial cells are of 3 types: the microglia, astrocytes, and oligodendrocytes. Microglia are the phagocytic cells that phagocytosize the microbes which enter the central nervous system from meninges and blood vessels. Astrocytes are found throughout the brain, joining the blood vessels and investing synaptic structures, neural bodies, and neural processes. Their important function is in the role of supporting the cells, neurons, transport mechanisms, inflammatory and reparative reactions, and also helps the formation of the blood-brain barrier. They also help in maintaining optimal concentration of the ions and neurotransmitters, especially glutamine in the brain neurons. Whereas oligodendrocytes, these are the cells that form myelin around within the cell, within the central nervous system. The axons in the central nervous system do not have Schwann cells. Orthodromic and antidromic conduction. Impulses normally pass in one direction only, that is from synaptic junction or receptor along the axon to their termination. Such conduction is called as orthodromic conduction. Conduction in the opposite direction is called as antidromic conduction. It is seen in the sensory nerve fibers supplying the blood vessels. These are the different neuroglial cells. See astrocytes, the oligodendrocytes, and the microglia. So microglia, they help in the phagocytosis, whereas oligodendrocytes, they help in the myelin formation. You can see the branches, and astrocytes, they provide nutrition, support, and helps in the formation of the blood-brain barrier. Nerve fibers types and their function. The nerve fibers classified based on Erlanger and Gassett classification. They have been divided into A, B, and C groups, and A group is further divided into A alpha, beta, gamma, and delta fibers. There is also numerical classification which is used for classification of sensory neurons, which is based on their origin of nerve fibers. There is also physioclinical classification. It has which has a clinical as well as physiological significance and is based on the sensitivity to hypoxia, pressure, and anesthetic agents. So this table shows the Erlanger-Gassett classification. You can see they've been divided into A, B, and C fibers. A further divided into A alpha, beta, gamma, and delta, where the classification is mainly based on the diameter of the nerve fibers and velocity of conduction. The diameter gradually decreases as from A alpha to C fibers, and the velocity of conduction is also going to decrease as they come from A alpha to C fibers. A alpha, they are proprioceptors and somatic motor. A beta, they carries touch, pressure. They are also motor fibers. A gamma is a motor to the muscle spindles. A delta carries pain, cold, touch sensation. Beta are the preganglionic fibers of ANS. C fibers, they are the they carry pain, temperature, and some mechanoreception and reflex responses, whereas C fibers, they are postganglionic sympathetic fibers. The numerical classification is on the origin, where 1A fibers, they originate from muscle spindle, that is annulospiral endings. 1B from Golgi tendon organs. 2 fibers from muscle spindle, that is the flower spray endings, touch and pressure sensors. 3 is they originate from the receptors of pain and temperature, and some from touch and pressure receptors as well as 4 from the pain and other receptors. There is a physioclinical classification where they are classified based on the sensitivity to hypoxia, pressure, and the local anesthesia. The B fibers, they are more susceptible for hypoxia, and the C fibers, they are least susceptible for hypoxia, whereas the A fibers, they are most susceptible for pressure. C fibers, they are most sensitive for the local anesthetics. This table gives the difference between myelinated and unmyelinated nerve fibers. In myelinated, multiple layers of Schwann cell membrane makes myelin formed by coiling of membrane many times around the axon, whereas in unmyelinated, axons are simply surrounded by the Schwann cells without a wrapping of the myelin. Myelinated fibers, they have a faster conduction of nerve impulses, that is almost 50 to 100 times than the unmyelinated. because of the saltatory conduction, that is the jumping of impulses from one node of Ranivere to another node of Ranivere. In unmyelinated, you can see the slower conduction of the impulses as it is a continuous process due to the lack of myelination. Some of the examples for myelinated fibers are all preganglionic fibers and the nerve fibers in the somatic nervous system, and for unmyelinated, all post-ganglionic fibers of autonomic nervous system and the nerve fibers in somatic nervous system less than 1 millimeter in diameter. Coming to degeneration and regeneration of the nerve fibers. When a nerve fiber is injured sufficiently, the part of distal to the injury dies. Some changes in the proximal part are also seen. The entire process is known as degeneration, which is also called as Wallerian degeneration, after Waller, who described it. Some of the common causes of injury are transaction, crushing injuries, injection of toxic or poisonous substance to the nerve, ischemia or interference in the blood supply, hypopyrexia, that is increase in the body temperature, can lead to injury. The grading of the injury is based on Sunderland. So they are called as first, second, third, fourth, and fifth degree injury. First degree injury is where it is most commonly seen and is secondary to ischemia caused by direct pressure to a nerve or for a limited time. Ischemia produces local anoxia with a temporary impairment of the nerve function. It gets corrected within hours or to few weeks because of the axon is not destroyed but merely loses its functional properties for a short time. Second degree injuries. It occurs because of the prolonged or severe pressure damages the nerve fibers at the pressure point and eventually causing the death of axon locally and distally. Third degree injuries where there is endoneural tubes becoming interrupted, and the fourth degree injuries where the fascicles becomes disorganized, and fifth degree injuries where through and through cutting of the nerve fiber occurs, that is in case of complete transaction. The degenerative changes occur at three levels: the changes that can be seen in the nerve cell body, changes that seen in the distal stump, and changes that seen at the site of injury. Series of degenerative changes. The change in the nerve cell body begin within 48 hours of injury. There you can see the chromatolysis, the Nissel's granules, they disintegrate. The Golgi apparatus, mitochondria, and neurofibrils are fragmented and they disappear. Cell draws more fluid and become round. The nucleus increase in size and move to the periphery. The changes in the distal lump begin within 24 hours of injury. The axis cylinder breaks into short segments and few days later and little debris is left. Then myelin sheath breaks up more slowly than the axon into small oily droplets. It occurs in two stages. The physical destruction occurs up to 8 to 10 days and the chemical destruction starts on the 8th day and goes till 32 days. The nucleus of the Schwann cell starts multiplying mitotically, which fill up the endoneural tubes. Macrophages from endoneurium starts phagocytosizing debris of the axis cylinder and myelin sheath. Once the debris is cleared, Schwann cells cytoplasm starts proliferating and it fills up the endoneural tubes. Changes in the site of injury, that is Schwann cell, differentiate into thin elongated cells, mainly from the distal end and very little from the proximal end. It can bridge up to the gap of 3 centimeters and the rate of progress of the growth is around 1 to 2 millimeters per day. There can also be regenerative changes. A degenerated nerve may regenerate provided there is a neurolemmal sheath. In places where the neurolemma is absent, there can be no regeneration. Nerve fibers of central nervous system, therefore, once degenerated, never regenerate as these nerves have no neurolemma. The degenerative and regenerative process go side by side. The changes in the nerve cell body begins at around 20 days and is completed within 80 days. The Nissel's granules and Golgi apparatus gradually reappear, cell regains its normal size, and nucleus returns to its central position. And the repair of cell may even occur if the axon does not regenerate. The changes in the distal stump and at the injury uh, is accordingly occurs. Axis cylinder from the proximal stump elongates and then grows out in all the direction as rounded pseudopods like structures called fibrils towards the distal stump. Each axon gives rise to 50 to 100 fibrils which are guided by strands of Schwann cells into distal ends of the endoneural tubes. 2 to 3 weeks after the nerve section, the distal endoneural tube contains varying number of developing fibrils. Eventually, all the fibrils reinnervating one tube degenerate except one which thereupon progressively gets enlarged to fill the tube. To begin with, the rate of growth is around 0.25 millimeters per day, but once it enters the distal stump, the rate of growth becomes 3 to 4 millimeters a day. As it grows deeper down, the rate of growth further increases because of the mechanical conditions for the regeneration are more suitable than those in a cut nerve end. In approximately 15 days, Schwann cell filling the endoneural tube starts lying down the myelin sheath around the successful fibril, which is completed by 1 year. Increase in the fiber diameter takes place very slowly, and the final diameter is attained is 80 to 85% of the normal. It is limited by the diameter of the distal tube and the size of the parent nerve cell. Therefore, functional recovery is not full, which takes a longer time and may be associated with a different complication if it does not reach its own cut stump. That is why, therefore, motor nerve recovery may be complete, but for a mixed nerve, it is rarely so. So this diagram showing the degenerative and regenerative changes. When the axon is severe, the distal part degenerates and it is called as Wallerian degeneration, and you can see some changes in the cell body also where the Nissel's granules they disintegrate. Okay, and the degeneration and regeneration they occur parallelly. The stump it starts giving pseudopods. There will be plenty, and one pseudopod or fibril will be able to successively penetrate into the neuronal cells. I mean, if the neural lemma is intact, okay, and which eventually grows and it is going to reach to the target end, and it completely regenerates. Degeneration hypersensitivity. When the motor nerve to skeletal muscle is cut and allowed to degenerate, the muscle becomes extremely sensitive to acetylcholine. This phenomenon is referred as denervation hypersensitivity or supersensitivity, and it is seen in smooth muscles also. Smooth muscles, unlike skeletal muscles, do not atrophy when denervated, but it becomes hyper-responsive to the chemical mediators that normally activates it. Hypersensitivity is limited to the structure immediately innervated by the destroyed neurons. For example, the response of the denervated iris. If the postganglionic sympathetic fibers to one iris are cut in an experimental animal, and after several weeks, if norepinephrine is injected intravenously, the denervated pupils dilates widely. As a much smaller, less prolonged response is observed in the intact side. Some of the causes for denervation hypersensitivity are synthesis and activation of more receptors. There is an increase in the area of muscle membrane sensitive to acetylcholine, and acetylcholine receptors of fetal gamma subunits appear in the orthograde degeneration, that is Valerian degeneration part distal to cut degenerates. Retrograde degeneration of the axons is towards nearest collateral. Okay. So this is diagram showing denervation hypersensitivity, where receptors becomes sensitive to circulating the neurochemicals, and there you can see the increased response because of the increased number of the receptors which are sensitive to a neurotransmitter. This is in brief about the neurons. Thank you.